Hello, this is Dr. Morris. I'm continuing the lecture series in microbiology. We'll talk today about viruses, viroids, and prions. This is chapter 13 in your textbook. In the early days of microbiology, the term filterable agents or filterable viruses, when we talk about the word virus, it derives from the word poison. It was used to designate an infectious agent that passed through the filters that retained bacteria. Now, later the term virus alone came into use. At that time, no one was sure what this particulate nature what the particulate nature of these submicroscopic organisms or agents were. Now, recognize the filters that had been set up previously clearly would filter out bacteria, but then to see a smaller agent, something that was smaller than bacteria, to be able to pass through and cause disease was significant in the development of microbiology. Viruses are obligatory intracellular parasites that require a living host in order to multiply. Now, this is much different than what we see in bacteria, because remember, bacteria can exist on the outside of a living host. It can exist on a fomite, an agent, or an inanimate object, but then it also can exist inside a human host as well. So with viruses, we see the term host range, which refers to a spectrum of host cells and the virus can infect. Viruses can infect bacteria. They are called bacteriophages or phages then, and in your laboratory study you've had an opportunity to look at bacteriophages or phages. Most viruses are much smaller than bacteria, although some larger ones approach the size of very small bacteria. Viral size range from about 20 to 1400 nanometers. A virion is fully developed complete viral particle. A viral nucleic acid may either be DNA or RNA in double-stranded forms. It may be linear or even in several separate segments. The protein coat is the capsid. It is made of protein subunit referred to as a capsomer. The capsid may be covered by an envelope in some combinations of lipids, protein, carbohydrates, molecular structures you're very familiar with at this point. The envelope may be covered by spikes projecting from the surface. Some viruses use these spikes to adhere to red blood cells, causing a clumping referred to as hemagglutination. Hemagglutination is a laboratory study that can be performed on blood serum to be able to identify whether viral elements are there. You look for the phenomenon of hemagglutination. If this is visualized, then you suspect that the tested microorganism is a virus. Viruses not covered by an envelope are known as non-enveloped viruses. Viruses can contain DNA and RNA. They also have a protein coat. Some are enclosed by an envelope. Some viruses have spikes. Most viruses infect only specific types of cells on one host. Host range is determined by the specific host attachment sites and cellular factors. Viruses may be classified into morphological types. Helical viruses resemble long rods. Their capsid is a long, hollow cylinder with a helical structure. Examples are tobacco mosaic viruses or bacteriophages M13. Polyhedral viruses usually have a capsid in the shape of an isohedron or a polyhedron, a 20 regular triangular faces. Examples are adenovirus and poliovirus. Enveloped viruses have their capsid covered by an envelope. They are roughly spherical, but no pleomorphic or variable shapes are seen. A helical virus is the case the helical capsid is folded, it's not extended in a rod-like form, such as influenza virus, which is referred to as an enveloped helical. A polyhedral virus, such as the herpes virus with a capsule, is an enveloped polyhedral virus. And here are some illustrations of various viruses. This is a helical virus that you're looking at in the illustration to the left. And then the Ebola virus is a typical helical virus, as you see on the right. This is a polyhedral virus. The mastinovirus is an example of that. The herpes virus, also another example. There are complex viruses such as the pox virus that do not contain identifiable capsids. They may have several coats around the nucleic acid, like many bacteriophages that have polyhedral heads or helical tails. As we look at the taxonomy of viruses, in this text we group viruses according to the range, that is, animal, bacteria, or plant viruses. Current classification systems are based on the type of nucleic acid morphology, the presence or absence of the envelope, and so on. A viral species is a group of viruses sharing the same genetic information or ecological niche. Important definition: the viral species, a group of viruses sharing the same genetic information and ecological niche. The suffix virus is used as the genus name, and the family name ends in viridae. Family names end in viridae, genus names end in virus. Viral species, a group of viruses sharing the same genetic information and ecological niche. Common names that are used for species. Subspecies are designated by a number. And we'll see many viruses that are labeled by number. The bird flu, for example, is labeled by a number now. And then we look at some examples of viruses, their names, and the numbering system. Herpes viridae, herpes virus. Human herpes virus type 1. Humans herpes virus type 2 and type 3. Retroviruses, lentiviruses, and human immunodeficiency viruses type 1 and HIV type 2. Now, growing bacteriophages in laboratories can be somewhat challenging. Bacteriophages require a specific host bacterium for growth. Growth medium for a host may be liquid or solid, but solid medians are used for detection and for counting viruses by plaque method. For this method, a melted agar suspension of a host cells and bacteriophages, phages for short, are poured into a layer over an agar on the surface of a petri dish. The bacteria develops a turbid lawn of bacterial growth, except for where it's been destroyed by proliferating phages. So as phages enter into a intrabacterial location, it will cause a destruction and leaves a clearing area. This cleared area is referred to as a plaque. The counts of the phages are made to determine the plaque-forming units. Viruses can be cultured in suitable viral animals, and some can be grown only this way. Because the signs of diseases of the animal are often significant, this method can only be used for diagnosis. Another method is using embryonic eggs, which can be inoculated by a hole drilled in the shell. Growth may be detected by death of the embryo or the formation of pox or lesions on the membrane. Most recently, cell cultures have been method of choice in viral cultivation. Animals or plant cells may be separated and grown as a homogeneous collections of cells, not unlike bacterial cultures. In containers, the cells contain and adhere to the surface to form a monolayer of cells. Cell infection by virus causes observable death and damage known as a cytopathic effect, or CPE, which can be used much as the plaques are for counting and detecting viruses. Primary cell lines are derived directly from tissue and tend to die after a few generations, but a few specialized human cell lines may be cultivated for 100 generations or so. Diploid cell lines developed from embryonic human cells are used to culture rabies virus for human diploid cell vaccines. They can be maintained for about 100 generations. Continuous cell lines, often cancer cells such as HeLa cells, can be maintained for an indefinite number of generations. So, just as a review, viruses must grow in living cells. Bacteriophages form plaques on a lawn of bacteria. Animal viruses may be grown in living animals or in embryonic eggs. Animals and plant viruses can be grown in cell cultures. Continuous cell lines can be maintained indefinitely. The most common methods of identification is referred to as serology, or serological studies. The virus can be detected and identified by its reaction with antibodies, which are specific proteins produced by animals in response to viruses. Again, as a review, the cytopathic effect, which you already recognize, and then serological test, or serology, detection of antibodies against viruses in a patient. For example, if you want to see if a person has been exposed to the measles virus before. You can check the antibody levels in that individual to see if they were previously exposed to measles. Chickenpox, the same way. Individuals who have received vaccinations, for example, vaccination for hepatitis B, you would obtain serology to see if the person has indeed developed antibodies to hepatitis B. The use of antibodies to identify viruses in neutralization tests, viral hemagglutinin, as I've already referred to, as when intracellular viruses exist inside red blood cells, it will cause the blood cells to clump together. And then the other studies, such as Western blot. Nucleic acids can be done before. We've talked about polymerase chain reactions before, PCR, as being able to detect and identify viruses based upon the genetic information. Viruses have only a few genes. Most enzymes encoded in viral nucleic acids are not part of the virion, but are synthesized and function only within the host cell. The viral enzymes mostly replicate viral nucleic acid and seldom the viral proteins, which are supplied by the host. Thus, the virus must invade the host cell and take over its metabolic machinery. The most familiar type of viral life cycle is the lytic cycle, or the T-even phages of T2, T4, T6. Now, the tail of the phages is adsorbed or attached to the host cell. This is a highly specific reaction depending on complementary receptors. The phage forms a hole in the cell wall using phage lysozymes to drive the tail core through the cell wall in penetration. It then injects the DNA into the virus into the cytoplasm. The head, or the capsid, remains outside. The viral DNA causes transcription of RNA from viral DNA and thus commandeers the metabolic machinery of the host cells for its own biosynthesis. And this is a key characteristic of what we see with viruses. For several minutes following infection, complete phages cannot be found. Thus, this is called the eclipse period. During the maturation period that follows the eclipse period, the phage DNA of the capsid formed separately are assembled into virions. When complete, the host cell lyses and releases these virions. The time required for a phage adsorption to release is called the burst time, about 20 to 40 minutes, and then the number released is the burst size. The stages of phage multiplication can be demonstrated with one-step growth experiment. So the multiplication of bacteriophages or the multiplication of viruses, these are important and you should be able to recognize these steps. This includes attachment, penetration, biosynthesis, maturation, and release. In attachment, we see the phage is attached by tail fibers to the host cell. In penetration, we see the phage lysozymes opens the cell walls, tail sheath contracts and forces the tail core in and then the DNA into the cell. Production and biosynthesis of phage DNA and proteins, and then in maturation we see the assembly of the phage particles and then release the phage lysozymes and breaks the cell wall again. And these are the steps that we see. There is a short video on your laboratory study about the HIV virus. I highly recommend that you look at that video. It goes through each of these steps, the attachment, the penetration phase, how these viruses replicate inside by using the DNA machinery, or I'm sorry, I should say the genetic machinery of the host cell. The maturation process and then the release process is illustrated. You can see also in a one-step growth curve the proliferation of these viral elements. The penetration phase, the biosynthesis and maturation phase, then the phage particles and release by lysis that takes place. The lytic cycle, again, just as a review, phages cause lysis and death of the host cell, and the lysogenic cycle, prophage DNA incorporated into the host DNA. The lysogenic phase and transduction, I won't go into a lot of detail about. those of you who are interested can certainly do additional reading of this material to fully appreciate this process. Now, multiplication of animal viruses, which is in part one of the areas that we're most concerned with because this involves human diseases, or human viral diseases. Multiplication of animal viruses follows the general pattern just described, but there is an important difference. Animal viruses have no tail, so attachment is by the spikes, and we've already talked about the spikes before. Penetration occurs by the fusion of the envelope with the hot plasma membrane or by a non-enveloped virus somehow entering into the cytoplasm. And again, I want to emphasize again, look at this small video clip that's a part of your laboratory study on the HIV virus, and you can see this stage fully illustrated or fully animated. Penetration occurs by a process of endocytosis, requires the cell's plasma membrane to fold inward and turn into a vesicle. Now, we described this process before when we talked about the cell membrane of the human cell in this process of endocytosis, where because the virus can mimic the coat of the bacteria, both having a phospholipid layer, will engulf the virus because it's a foreign particle that sits on the surface and will bring it into the cell. There's another alternative method referred to as of penetration, referred to as fusion. This is where the viral envelope actually fuses with the plasma membrane. It releases the capsid into the host cell cytoplasm. Now, once inside the host cell, so once the virus is inside the cytoplasm, the viral nucleic acid separates from one protein coat in a process referred to as uncoating. So again, multiplication of animal viruses, we see attachment, virus will attach to the cell membrane, penetration by a process of endocytosis or fusion, and then uncoating by viral or host enzymes, and then biosynthesis, production of nucleic acid and proteins, and then nucleic acid and capsid proteins will assemble, and then the release by budding the enveloped viruses or rupture. The viral envelope fuses with the plasma membrane and releases a capsid into the host cell's cytoplasm. Once inside the host cell, the viral nucleic acid separates from the protein coat in this condition. We'll see in the multiplication of DNA viruses, it can occur entirely in the cytoplasm. We see this occur in the pox viruses, or the DNA may be formed in the nucleus of the protein in the cytoplasm, the final assembly taking place in the nucleus. This is the process of attachment, penetration, and uncoating that we see as the animal virus infects the human. The steps are as follows: attachment, endocytosis, penetration, and uncoating. The release of enveloped viruses by budding. Multiplication of the DNA virus. Again, these steps I just go through briefly. Those of you who are more interested or want to go further in depth, please do so and refer to your text for additional reading and supplemental material. Pathways of multiplication of RNA-containing virus, as you can see, involves multiple other steps. We still see the attachment, penetration, and uncoating process, but then we'll see RNA replication take place, aspects of translation and synthesis of viral proteins before we see the maturation and release. And again, those of you who wish to go into more detail can certainly read that in additional supplemental material inside the textbook. Now, retroviruses are an important aspect and an important feature that I'd like to bring up just a bit. When we talk specifically about retroviruses and how they penetrate the cell, you will see the process of how the virion penetrates the cell and its DNA is uncoated. You'll see a new viral DNA transported into the cell's nucleus. It's integrated as a provirus, and you do need to recognize the term provirus. Proviruses may divide indefinitely in the host cell. This is a sample of HIV disease. Because these viruses can divide indefinitely in the host cell, and because, specifically like with HIV disease, its predilection is towards cells of our immune system, because of this indefinite proliferation, it completely destroys the individual's immune system. And remember, it's competing for the same energy source as normal cells would, and thus you get significant compromise to the host. Transcription of the provirus may also occur, producing RNA for new retrovirus genomes, and the RNA that encodes the retrovirus capsid and envelope proteins. Mature retroviruses leave the host cells, acquiring an envelope as it buds. Now, the other important aspect of the retrovirus is its ability to mimic a lot of the machinery of the host cell, and using the host genetic machinery or metabolic processes to be able to carry out this process of replication. Adenoviridae cause some common colds. The term is derived from adenoids, but adenoviridae cause many other forms of common cold. Poxviridae, infections such as smallpox. Pox are pus-filled sacs on the skin, is where the name is derived. Herpesviridae, named after spreading of herpetic appearance of cold sores. Official name is human herpes virus, or HHV. Also has a numbering system. The numbering system is important to identify the location of the organism. These are the common forms of the herpes virus or herpesviridae. As you can see, the number system 1 through 8. Herpes simplex type 1 is a simple cold sore. 90+% of the human population has this virus. Certainly has the antibodies to this virus, I guess it's better said, has the antibody to the virus, but if I can say it, but may not present with symptoms. Herpes simplex type 2 is genital herpes. Typically you see it on either the, you see penile lesions, or you may see lesions along the, around the vagina in the labia majora or labia minora. Also herpes simplex type 3, which is varicella or chickenpox. The herpes human virus type 3, varicella, is a common childhood disease. HHV4 is Epstein-Barr virus. 5 is cytomegalovirus. 6, not seen too often, roseovirus. And then 7, mostly affecting infants. And then number 8 is the cause of Kaposi sarcoma, which can also be seen as well in HIV patients. Papovirus, named for the papillomas or the warts. Polyomas or tumors, or vacuolation, which is a cytoplasmic vacuole produced by some of these viruses. This is a DNA virus, such as the Papoviridae, replicates in the nucleus of the host cell. Basically, protein is synthesized by transcription and translation in the conventional matter, similar to bacteria. When cells multiply in an uncontrolled way, the excess tissue is called tumor, which is malignant, and it can be cancerous and benign if not. Leukemias are not solid tumors, but an excess production of white blood cells, whereas sarcomas, chicken sarcoma, connective tissue type of cancer, adenocarcinoma, a glandular tissue that can be transmitted by viruses. The adenovirus, herpes virus, pox virus, papovirus groups all contain oncogenic viruses. Among the Papoviridae or viruses are papillomaviruses that cause warts. Herpes virus, including Epstein-Barr virus, may cause Burkitt's lymphoma and nasopharyngeal carcinomas. Heptenovirus, also causing hepatitis B, also known as the role of liver cancer. RNA oncogenic viruses, only the retrovirus seem to be oncogenic among the RNA types. Their oncogenic activity seems related to the production of reverse transcriptase, DNA synthase, and viral RNA becomes integrated into the host cell's DNA, such as in the provirus, which I alluded to earlier. In some cases, this may convert the host cell into a tumor cell. So recognize, only the retrovirus seems to be oncogenic among RNA types. Now, activated oncogenes transform normal cells into cancerous cells. Transformed cells have increased growth, loss of contact inhibition, and I'll explain that. Normal cells in the body, when they come in contact with one another, will stop growing. This is called contact inhibition. This is why we don't get too many cells. We have stem cells in our body that will continually produce new lines of cells. However, because of contact inhibition, it restricts the growth of the cells. The cells touch each other and growth will stop. When we lose contact inhibition, then cells will start to grow out of control. So cells that form cancer are cells that do not have contact inhibition. And because of that, those cells will grow out of control. They'll stack upon each other and develop tumors. Tumor-specific transplant and T antigens also seem to increase the growth. Genetic material of oncogenic viruses become integrated into the host cell's DNA. We've talked about this in other aspects of, or other types of cancers. Oncogenic DNA viruses. Here are the lists of some of the common type. Adenoviridase, herpes, that should be herpes, I'm sorry about the typo, herpes viridae, pox viridae, papavirinae, and then hepanoviridae. Oncogenic RNA viruses, the most common type is the retroviridae. Viral RNA is transcribed to DNA, which is then integrated into the host DNA. HTLV1, HTLV2 are the most common type. So again, you can have DNA viruses that cause cancer. You can have RNA viruses that can cause cancer. A number of DNA viruses exist. Typically for RNA viruses, only the retroviridae. Now, some other terms, latent viral infections. We've already talked about latent viral infections, or we will talk about it, I'm sorry, in epidemiology. But latent viral infections, sometimes a virus can remain latent. In other words, it can exist in the body. Typically, in most of the cases that we're talking about, typically the virus will lay dormant on a nerve cell of the host for long periods of time without causing disease. This is referred to as a latent infection. Stress or some other factors that can trigger the reappearance of these diseases. This is the case in the herpes virus, which causes cold sores, and chickenpox virus, which causes shingles. Now recognize that again, these viruses will exist in our body and will lie dormantly on a nerve cell. And then when the right environmental settings or the right conditions in the body, as we said here, can sometimes be triggered by stress, we see re-exacerbations of the conditions, herpes and shingles. So just as a review, latent viral infection, virus remains asymptomatic in the host cell for long periods of time. Cold sores and shingles. Now a persistent viral infection, disease processes occurs over long periods of time. This is generally fatal. There is a term referred to as subacute sclerosing panencephalitis. Subacute diseases lie somewhere between the acute phase of a disease and a chronic phase of a disease. Now, the term persistent viral infection refers to a disease process that occurs gradually over long periods. Originally called a slow viral infection, the term refers to as a slow progress in the disease. An example is subacute sclerosing panencephalitis, in which measles virus continues to reproduce slowly, causing this rare encephalitis. A number of neurological diseases called spongiform encephalopathies may be caused by prions coined as proteinaceous or infectious particles, which have characteristic unique to biology. Now, prions. This is the prototype of these diseases is scrapie or a neurological disease of sheep. Prions appear to be a pure protein that lack nucleic acid. Among the hypotheses to explain their reproduction is that a prion protein is a gene found in a normal host DNA, and the abnormal form of the protein causes the disease condition. Another possibility is that the prion may contain an undetectable amount or small amount of nucleic acid. Diseases caused by this agent, prions, other than scrapie that you see in the sheep, they include mad cow disease, which gets a lot of media play, also known as Creutzfeldt-Jacobs disease, also known as Kuru or Gerstmann-Straussler-Scheiker syndrome. That's a tongue twister. These diseases, or this disease, and all these names relate to a disease that infects primarily the central nervous system, and accordingly causes a poration or poor appearance in the nervous tissues, particularly the brain, and leads to a pan of neurological deficits. The disease caused by the conversion of normal host glycoprotein into an infectious form. A prion infection produced by cells is secreted in the cell surface. During the infection, the infectious prion reacts with the normal glycoprotein on the cell surfaces and then converts it into an infectious form. The infectious form is then taken up by the cell in a process of endocytosis and accumulates in lysosomes in the cell fragments and then accumulates in the brain, forming plaques. These plaques are important for diagnosis but do not appear to be the cause of cell damage. The infectious proteins, inherited by transmissible or ingestion, transplant, or surgical instruments, in spongiform encephalopathies such as sheep scrapie, Creutzfeldt-Jacobs disease, Gertzmann-Straussler's-Scheiker syndrome, fatal familial insomnia, and/or mad cow's disease. Those are the chemical symbols that you see related here are related to the normal cellular protein, prion, and then the Scrapie's protein. Again, I'm not holding you responsible for that, just to know the concepts of the disease. And now you can see in the illustration before you now, it's just the little steps of how these little portions of infected proteins can enter into a system. In plant virus, they can enter through the wounds or via an insect. Viroids are infectious RNA, such as potato spindle tuber disease. Again, as I told you previously, I don't focus a lot on plant disease. We're here primarily to recognize human disease. Virus families, single-stranded DNA, non-enveloped viruses. Again, they're DNA viruses and RNA virus. Parvoviridae, human parvovirus, Fifth disease, anemia in immunocompromised patients are forms of viral elements that can cause this. The Hypnoviridae, named because of hepatitis, contains DNA. They differ from other DNA viruses because they synthesize DNA by copying RNA with reverse transcriptase. Now, if you recall, we said reverse transcriptase we see in RNA viruses, so now we're talking about this in a particular DNA virus. Mastinovirus, respiratory infections in humans, can cause tumors in animals. Papillomavirus cause human viral warts. Poliomavirus cause tumors and can even cause death. Arthropox virus, vaccinia, and the smallpox virus. Malusi-pox virus, smallpox in molluscums. You can also see it in cowpox as well, pox viridae. And then some of the double-stranded DNA viruses include the simplex viruses, human herpes simplex virus, varicella virus, lymphocryptovirus, cytomegalovirus, roseolavirus, human herpes simplex virus type 7, Kaposi sarcoma, which is type 8. Some of these herpes viruses can remain latent in the host cells, as we've talked about earlier. Hepatinovirus with hepatitis B virus uses reverse transcriptase to produce DNA from RNA. Again, this is a DNA virus that has reverse transcriptase. Single-stranded RNA viruses, enterovirus include poliovirus and Coxsackie virus, rhinovirus, and hepatitis A virus. Hepatitis E virus, or the Norwalk agent, causes gastroenteritis. Alpha viruses are transmitted typically by arthropods, which can occur in many of the encephalitises that we see. Encephalopathies, I guess, is a better term to be able to use, that occur. Inflammation in the lining around nervous tissue. Western equine and eastern equine encephalitis are examples of those. Arboviruses, which replicate inside the arthropod, include yellow fever, Dunge fever, SLE, and West Nile virus, St. Louis encephalitis, and then hepatitis C virus. Corneal virus typically is the cause of upper respiratory infection. Fasciculoviruses and lysoviruses, which is the rabies virus, can cause a number of animals' diseases, also known as rhabdoviridae. Enveloped are helicoviruses, filoviruses, or filoviridae. The Ebola and Marburg viruses are common types of organisms that you can see with these infections. Perinomyxovirus, morbillovirus, perimyxovirus is all cause parainfluenza, mumps, Newcastle's disease, so forth. Hepatitis D virus depends on a co-infection with the heptenovirus or the delta virus, then the influenza virus, virus type A and B, influenza C, develop spikes, also can agglutinate RBCs, and again, as I alluded to earlier, hemagglutination is one way to identify in a blood or a serum specimen whether or not viral elements are there, because the spikes that exist on the surface of the viruses, they cause an agglutination or these red blood cells to clump together and can be diagnostic of viral infections. Buniovirus, hepnovirus, these are single-stranded RNA viruses and multiple RNA strands. Arenovirus, these are helical capsids, RNA-containing granules. Lymphocytic choriomeningitis, the Lassa fever that also gets media attention from time to time when exposed in the community. Arenoviridae are common organisms. The lentivirus category is important because the lentivirus is the human immunodeficiency virus, or HIV disease. This is an oncogenic type of virus. In the category of oncogenic viruses, they use reverse transcriptase, and remember that's a key enzyme. We talk about oncogenic cancers, or oncogenic viruses have a reverse transcriptase to produce DNA from the viral genome. It includes all RNA tumor viruses. Retroviridae is the category. Reoviruses, respiratory enteric orphan, rotavirus, mild respiratory infections, and also gastroenteritis. We can see this a lot, especially in developing countries, rotaviruses, and we also see this periodically in the U.S., a cause of gastroenteritis. Colorado tick fever, all